The broadcast is now starting. All attendees are in listen-only mode.
Good morning, good afternoon, and good evening, everyone. And thank you for joining us for today's webinar. Today's talk is entitled, Why Do So Many Phase 3 Clinical Trials Fail? My name is Kelsey Brown, and I am the Director of Medical Writing Solutions at TransPerfect Life Sciences based in London. And it's my pleasure to be your moderator for today's session. And today's event will run for approximately 60 minutes, and that'll include some time at the end for Q&A with our speakers. The session is designed to be interactive, so it really works best when you're involved. So please feel free at any time to submit your questions or your comments for our speakers by using the questions chat box in your control panel, and we'll get to your questions during the Q&A session. At this time, all participants are in listen-only mode, so if you do require any assistance, just pop me a message in the control panel in that messaging tab that you have there. And please note this session will be recorded and it will be made available to you on the IDDI website for viewing within 24 hours after the webinar is complete. The event is proudly hosted by the International Drug Development Institute, IDDI. IDDI is an expert center in biostatistical and integrated eClinical data services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI is headquarters near Brussels, Belgium, with the U.S. Center of Operations in Raleigh, North Carolina. So with that, I will introduce our two speakers today. We have Dr. Mark Busey, the founder and chief scientific officer at IDDI, and Dr. Everard Assad, medical director at IDDI. So thank you very much for joining us, and I'll just pass the ball over to you.
Thank you very much, Kelsey. Hello to everyone, and thank you for joining our webinar. I'm waiting for Mark. Yes, thanks, Mark. So this is the story for today. I will play the CMO in a biotech company, and I am planning to move to phase 3, but I have some concerns, and I will describe what can be called the epidemiology of failure. And I will ask Mark, how can I reproduce in phase 3 the good results that I have in phase 2 with my drug? And Mark will be the statistical consultant who will give us notes of caution, describe main statistical reasons for failure in phase 3, and will give us some advice going forward in new studies. Well, Mark, as you know, oncology is well known for failures in drug development, arguably more so than other areas, although this may depend a little bit at what's looked at. But there are many ways to describe this, but if we just look at the left-hand panel, these researchers looked at 112 phase 3 trials that were published. And of course, this is a published literature, so it's not the whole of the existing studies, and it's only for 3 journals in a relatively small period, but slightly over half of them were negative studies. And these were studies of targeted drugs, which are usually more promising in oncology. So that by itself is reason for concern. Many phase 3 trials fail in oncology. And another look of this is shown on the right-hand side, where there are failure rates or success rates, if you will, of different transition phases in drug development. And there's a comparison of oncology, which is the light gray color, versus non-oncology, the dark gray. And it's pretty clear that the phase 2 to 3 transition is the one with the lower success rates, and apparently more so in oncology than in other areas. So this by itself is reason for concern. We can ask ourselves why, and there are, I guess, many reasons like these authors compiled for us. Some of them have to do with basic science related to the drug. Some have to do with pharmacology, let's say, or dose selection. Some are operational. but clearly many are statistical in nature, and I've highlighted some of them here. Some relate to trial design. For example, is the patient population in phase 3 perhaps different? Is the endpoint to be blamed in a way that I will describe? Also, so-called false positive phase 2 trials, why are they not often reproduced in phase 3? Are we overly optimistic in our assumptions? So this is the kind of thing that I want to explore with you in the next hour to better understand how to move forward. So the first thing is we know there is a population issue in the sense that if one compares response rates in phase 2, which are on the x-axis on this graph, with the response rate in phase 3 for the same agent or combination, we see that there is not a perfect correlation in the sense that there is a trend for a higher response rate in phase 2 as opposed to phase 3. On average, this delta in the differences in ORR is about 10%, which means probably that the population is changing when we go from phase 2 to phase 3. This is not unexpected. Typically in phase 2, we enroll patients who are perhaps more fit and sometimes have slightly different profiles than in phase 3. So we know this exists. What else could be at play? Incidentally, we know that oncology is the prime specialty where FDA, for example, grants accelerated approvals, so much so that 2/3 of all accelerated approvals in a certain time period were granted to oncology drugs. We also know that from 92 to 2017, FDA granted 93 accelerated approvals, and the ratio of regular approvals is to 174. So about 1/3 of all approvals the FDA has granted recently are accelerated approvals. So of the 93 accelerated approvals, the vast majority were based on single-arm trials. 72% of the studies were single-arm. So there may be an issue with trial design, also with the endpoint. As we see on the graph, the vast majority of cases had response rate as the endpoint for accelerated approval during this time period that is shown on the graph, on the slide. And so we may wonder about the population, about the single-arm issue, the endpoint issue. There may be a more general trial design issue whereby the expected benefit isn't always confirmed. If these authors have compared a very large number of randomized trials published over a 5-year period in 10 journals, and if we look, of course, at the positive trials highlighted in black here, there is a nice correlation between the expected benefit and the observed benefit, but a large proportion of studies in oncology are neutral in the sense that they don't favor any arm, or truly negative in the sense that they favor control. So there may be a general issue with expectations versus observations in treatment effects. And on the next slide, we see that this is confirmed, for example, in this other study, very interesting, where the authors paired 57 randomized phase 2 trials with their follow-up phase 3 studies, and they plotted the hazard ratios for either overall survival or progression-free survival, whatever was more relevant for each trial. And again, we don't see a perfect correlation. We see that as a general rule, the hazard ratio in phase 3 is closer to 1 than in phase 2, so much so that the ratio of hazard ratios, which would in theory be close to 1, is in fact far from 1 in the sense that the hazard ratio is typically lower in phase 2 than in phase 3. By that I mean further away from 1. So this is all well known, and the final slide I wanted to share with you, Mark, is, well, maybe there is a design issue of going from a single arm phase 2 to a randomized phase 3 study, but what about the cases where we have even a randomized phase 2 trial of a decent size, let's say here about 130 patients, and very nice results as far as the separation from survival curves, and then this is not reproduced in phase 3 without there being an apparent big change in population or in other features of the study that could readily explain this discrepancy. So what do you make of it?
Thank you, Eduardo. Yes, thanks for the question, and it's a complex question. Obviously, I don't have a single answer, but I would like to go through some statistical explanations, perhaps not of the full, extent of the problem, but at least that can explain some cases of failure. And there are really two main classes, and I will focus on the first one, which is called, variously optimism bias, or which is a form of regression to the mean. It's also sometimes known as the winner's curse. And I will, give you some examples of that. And essentially, the cause of the problem is that only successful phase 2 trials lead to phase 3. And some of these phase 2 trials may well be negative trials that in fact should have been negative, but were falsely considered to be positive. So in a sense, what you look at is the tail of the distribution. You look at the better tail of the treatment effect, and you think logically that the effect is larger than it truly is. And it can be shown, and I will go into the details of why, that the treatment effect in phase 3 trials are expected statistically to be smaller on average than in the preceding phase 2 trials. So this is a phenomenon that can be explained purely on a statistical basis. Nothing to blame, the trials were well done, the investigators did their job, but statistically, because you select the phase 2 trials that succeed, in fact, the confirmatory phase 3 trials will, on average, show less effect than the phase 2 trials that preceded them. And of course, this is, excuse me, this is of course only an average conclusion. You can't say anything about an individual phase 3 trial versus its counterpart in phase 2, but on average, you need to expect to be disillusioned. That's a problem with statistics, is that, in general, statisticians tend to be pessimists, and for good reasons that I will explain. The second cause of problem, I think you mentioned, some of the issues with single arm designs and stuff like that. We won't go into that because this is trivial. If you do a single arm phase 2 trial, then you have no idea about the treatment effect, in fact, or you have a very, unreliable idea. You might have a high response rate that is due entirely to the patient selection. So we're not even going to talk about single arm phase 2 trials. They are not reliable, and in general, I would certainly not advise people to do them in general, but we will leave that aside for today. What we have in addition is a lot of, something that we call the hacking bias, and it's well known to all statisticians. A phase 2 trial has been conducted, typically hugely expensive trial. It has to show something. So even if it's flat negative, people will look at subgroups, they will try to extract some information, and no blame again. I mean, it's human nature to find something positive to say. Even if you have no good news on the whole, you will try to find some good news that's hidden as a gem in the mud of negative results. And so that is completely comprehensible, but we need to be aware of it. Sometimes also people relax the significance level for phase 2 trials, and there are various schools of thought about this. Personally, I don't advise for this. If possible, again, we can discuss that, but I won't go into this initially. I will go back to the significance level to be used at the end of the talk to show that an alpha level of 0.10 is certainly not a good idea if you can do something better. So what about this winner's curse? The term, to be honest, I didn't know this term until I looked at the literature for explanations of this regression to the mean. And I was interested to see that as long ago as 50 years ago, this winner's curse was discovered not in clinical research, but in a completely different activity, which is, oil extraction and the purchase of oil fields by oil companies. So typically back then, these oil fields were bought in auctions, And in fact, so there were several bidders. And if you look at a situation here that I've depicted, you know, sort of schematically, suppose there are many oil leases or leases to be bidded for, and so there are several bidders. Suppose the intrinsic value of a specific field is around about $10 million, and now you have successive bids for that field, for the purchase of that field. And as you can see, of course, by definition, the bids can only go up. And so when you reach the value that, you know, intrinsically is the value of this field, and everybody sort of knows that that's round about what the value should be, if you really want to win, I mean, you've got to go beyond it. And so obviously, on average, the winning bid is going to exceed the intrinsic value of the field, because if everybody agrees that the $10 million is the price, no one will stop before $10 million, and a few people will be willing to go over. So that's the winner's curse. You win the bid, but you know that you've paid too much. Now, of course, what does this have to do with phase 2 clinical trials? Well, it's the same phenomenon. You bias your bid, in fact, and you select the higher bid. Likewise, if you do a phase 2 clinical trial, because you will only consider the results of your trial if it's very exciting, very positive, you actually select a biased sample of phase 2 clinical trials, which will go on to phase 3, and therefore the regression to the mean effect will play. So the regression to the mean was actually introduced by Francis Galton a long time ago, more than a century ago, well, more than a century ago. And Galton called it regression to mediocrity, which wasn't a particularly nice word, but that's how he termed it. And he showed that, in fact, adults are closer to average height than their parents. Think about that. It's actually a phrase that I find very difficult to understand, but you need to sort of read it several times and think about it. Stephen Sen, who's a very thoughtful statistician, has in fact shown an example of, you know, what might be considered regression to the mean. And I think this is a beautiful example that shows the phenomenon. Suppose that you are interested in looking at the diastolic blood pressure of patients at 2 different times. And suppose we call the first time baseline and the second time the outcome time. So assuming that these patients, you know, will have a change in their blood pressure in the meantime. And suppose, just for the matter of argument, that these patients receive nothing, and so therefore their blood pressure doesn't change. So we have 2 independent values on the same patients, but 2 independent values of the diastolic pressure at 2 different times. Now, suppose that you're interested in hypertensive patients. So patients who here, I've assumed, have a high blood pressure, and by high I mean more than 95 millimeter of mercury. So you take these patients, you select that population of patients, they have a high blood pressure, and those are the patients that you're interested in. Now, if you look at this graph, in fact, if you considered the baseline blood pressure versus the blood pressure at outcome, some of these patients were hypertensive to begin with, and their blood pressure has gone down. So even if these patients have received nothing at all, in fact, their blood pressure will be better, apparently, than it was initially, even though they haven't been treated and their blood pressure truly hasn't changed except by random variation. And of course, this is a biased graph. It only shows patients who were initially hypertensive, and it didn't show the patients who were initially normotensive, so who had a blood pressure below 95, and who in fact had an increased blood pressure, you know, at the outcome time. So in fact, by selecting again the patients who did poorly initially, these patients seem to do better over time just because of the regression to the mean. And that's exactly what's happening in phase 2 versus phase 3 clinical trials. In fact, some statisticians have studied this in detail, and there is a very interesting paper by Van Zwedt and colleagues in Significance, which is a statistical journal, but a pretty readable one. What they introduced in that paper is a notion called the exaggeration ratio. They say the exaggeration ratio is in fact a result of the regression to the mean, and it's the ratio of the observed treatment effect to the true treatment effect. And what's even better is that they can estimate rather the exaggeration ratio. So suppose we have done an experiment, a phase 2, let's say, we have the observed value of the treatment effect and its standard error. The ratio of these quantities is called a Z value. It's a standardized normal deviate. And suppose we have in parallel to that a database of trial results. And these authors have chosen to use the Cochrane database of systematic reviews, which of course is not relevant to the particular trial, phase 2 trial that we're looking at, but it's the database of all randomized trials that have been conducted across a number of indications. So you might say, well, this is not an appropriate basis to look at, but anyway, let's use it just for the sake of argument. This is 24,000, more than 23,000 clinical trials, and they have a distribution of the hazard ratio or of the treatment effect in all of these trials, and they use that as in fact the prior distribution. I will come to that in just a moment. But the point is, looking at this exaggeration ratio, before I show you the example, the point of this exaggeration ratio is that if you look at the Z value that corresponds exactly to a 0.05 significance level. So that would define the point under which, or the p-value under which you begin to be interested in the results of a phase 2 trial, because they are so-called statistically significant. We can draw the vertical line here, and the exaggeration is such that, and this is based on observed data from the Cochrane database. In fact, if you take 0.05 as your p-value of interest, in fact, the exaggeration ratio predicts that 25%, you have a 25% chance that the observed effect in your phase 2 trial is in fact more than 3 times larger than the real effect in that trial. Now that's pretty discouraging, because that means you really overestimate your treatment effect. There's a 50-50 chance that the observed effect in your phase 2 trial is greater than 1.5 times, in other words, 50% larger than what it should be. And there is only about a 25% chance that your treatment effect, the observed treatment effect, is round about the true value. That's the point here. Now, if you use the exaggeration ratio and adjust for it, as I will show in a moment, then in fact things are much better, because for the p-value of 0.05, now you only have a 25% chance that the observed effect is 2 times larger, which is much better than before. And there's a 50-50 chance that the observed effect is round about equal to the true effect. So the adjustment for the exaggeration ratio corrects for it in a manner that seems to work really well. And let's see how that plays out in an actual trial, a phase 2 trial of ruxolitinib. And this was a trial of a drug, this new drug here, that was combined with capecitabine in patients with metastatic pancreatic cancer for whom therapy with gemcitabine had failed. So they had received capecitabine, and this capecitabine-based regimen was tested. They compared ruxolitinib plus capecitabine versus the control treatment of capecitabine alone. About 127 patients. The hazard ratio was 0.79, not significant for survival, 0.75, not significant for progression-free survival. Negative phase 2, right? So why don't we move on to some other drug? No, these authors actually looked at subgroups. And we all know the dangers of subgroups, but anyway, this was a pre-specified subgroup. Well, that perhaps makes it a bit better, and it's a subgroup of patients with inflammation. In that subgroup, they had a hazard ratio of 0.47, and that was significant at the 0.01 level. So the confidence limits of this 0.47 hazard ratio were 0.26 to 0.85. And now we can use, in fact, a web application that Van Zwet and his colleagues have developed. So you can go on the web and type in this address, and you will be put in this application where you can choose the measure of effect. Here it's the hazard ratio. You introduce the lower 95% confidence limit, 0.26, the upper 95% confidence limit, 0.85. You press the run button, and this is what you get. What you get is 3 curves, right? One curve is the likelihood of the data. The black curve is exactly reflects this confidence interval here. It's a normal distribution with that confidence interval here. That's the likelihood of the data in the phase 2 trial that you've conducted, or that they conducted. Then you also have the blue curve here, which is this skewed distribution. It's right sort of centered around 1, which is no treatment effect, a hazard ratio of 1. But as you see, it kind of has a very heavy tail towards a significant, not a significant, but a positive effect, a lower than 1 hazard ratio. So that means a number of trials had showed a treatment effect, and then of course there were trials that showed no effect or a negative effect, and that's called the prior distribution of the hazard ratio, based again on the Cochrane database of many trials. And so using the data, or the likelihood of the data, and the prior distribution, we can use a Bayesian calculation to derive the posterior distribution, which is the red curve here. And the posterior distribution, as you see, is in fact similar to the data but shifted towards the prior. So it's as if you adjusted your observed data based on the fact that your prior distribution was in fact much less impressive than what you've observed. And so when we do this in the subgroup of patients with inflammation, remember you had the 0.26 to 0.85 confidence interval. If you adjust this using the correction for exaggeration, your adjusted hazard ratio is 0.62, and the credible interval, which is the Bayesian equivalent of the confidence interval, now goes from 0.33 to 1.02. So in a sense, you could say, well, in a sense, it's no longer significant. I know that this is a Bayesian approach, so you don't talk about significance anymore, but even so, you might think of this as being, you know, much less impressive than it was initially. And these investigators didn't do this. They thought that 0.01 was enough. They did the phase 3 trials in an enriched population, so only patients who had inflammation at baseline, and in fact, the trial was flat negative. This is a trial that should not have been done because the prior information wasn't, you know, or the phase 2 information, I should say, wasn't promising enough. So when we go back, Eduardo, to the list of reasons that you showed before for failure, in fact, even though everything is perfect, we can't avoid this statistical. So even if we have everything perfect, we can't avoid this phenomenon that you circled before, or that you highlighted before, which is that, you know, we have an over-optimistic assumption. We have an rather over-optimistic readout of what the true treatment effect is. We overestimate the treatment effect typically in phase 2 trials. That's the sort of bottom line from all I've said, is we know that on average, your phase 2 results are overestimated. That's bad news, but it's a fact.
Okay, well, thanks for that, Mark. It's a lot of food for thought, huh? Particularly the issue of the overestimation and the regression to the mean. Also, if you can, after this question I'm going to ask you now, if you can come back to Bayesian concepts, because you mentioned many words that I've heard in the past, but they're not very clear to me. I hope you can come back to the Bayesian explanations. But before that, how about p-values? You mentioned, you know, p-values, and in some of the examples of positive phase 2 trials that are followed by a negative phase 3, the p-values in phase 2 are very good, and you know, statisticians tell us that this is, let's say, the risk of a false conclusion, right? So how can you explain that?
Okay. Yeah, I mean, I think there is a big misinterpretation of p-values. You know, many studies, as we've seen, cannot be reproduced. And this is a paper, very interesting paper by John Ioannidis, who is in Stanford. And he has written a lot about the so-called replication crisis. And here you see, you know, one of his papers, an abstract from one of his papers, where he shows essentially that only 25% of results remain largely unchallenged if you take the original clinical research study and then the follow-up study that tried to replicate it. Only 1/4 of the studies can be replicated in his sample. But the causes of this replication crisis, I've already talked about regression to the mean. Of course, there are other causes. For example, the pressure to publish. You know, people will want to publish positive results at all costs, and so they kind of bias the published results positively. We all know that. There is also questionable research practices. We won't even go into that. People publish false results. But then there is this misinterpretation of p-values that I would like to go into from a statistical point of view. So what is a p-value? As you know, it's defined as the probability of observing a treatment effect at least as large or as extreme as that actually observed under the null hypothesis, i.e. if there was no treatment effect. So if there was no treatment effect, then if my p-value is 0.05, that means if there was no treatment effect, I have 5% chance that my results are in fact as extreme as those that I have observed. So that's kind of interesting, but I will show that it in fact makes a very unrealistic assumption and that it may be very misleading. I make the assumption that there is no treatment effect and then I try to reject that assumption, right? That's the point of a p-value. But that doesn't really quantify what we are truly interested in, as I will show, because the p-value has nothing to do with the magnitude of the, I'm sorry, I didn't say that. It has something to do with the magnitude of the treatment effect, but in and of itself, it does not quantify. If you don't tell me also the sample size of your trial, the p-value in and of itself does not reflect alone the magnitude of the treatment effect. It reflects it partly, but I need to know the sample size to go beyond. It's not sufficient either to quantify the probability that the next trial will succeed, and yet that's exactly what I'm interested in. If I have a p-value of 0.01 or 0.05 or something else, I want to know if that p-value informs me as to the probability that the next trial will succeed. And the p-value doesn't do that, as I will show. It's not the probability that the null hypothesis is true. You know, if I have a 0.05 p-value, that doesn't mean to say there is 5% chance that the null hypothesis is true. And I will go into the Bayesian framework because in the frequent risk framework, the null hypothesis does not have a probability. It's a fact that I assume. I say under the null, and I decide that the null is true or not. So it's something that I manipulate in the frequent risk framework, not so in the Bayesian framework, where I try to assume a certain probability for the null or the alternative being true. So that's why the Bayesian approach is really interesting for this particular problem. And finally, as I will also show, the p-value does not imply that the odds in favor of HA, the alternative hypothesis, relative to H0, the null hypothesis. In fact, the p-value of 0.05 does not imply that the odds are 19-to-1 in favor of HA relative to H0. So that doesn't mean to say that the alternative hypothesis is 19 times more probable than the null. And again, when I say more probable, I'm talking in Bayesian terms. So in order to understand this Bayesian business here, we can just look at the clinical trial as a diagnostic tool. That's essentially what we try to do. We want to show that the treatment works. We want to establish if the treatment works. And in fact, the clinical trial is a diagnostic tool for that truth that I don't know. And so the idea of a clinical trial is to run a trial to in fact make a difference between no treatment effect, which is called H0 or H0, and some treatment effect, which is the alternative HA, and then to run the trial and to decide based on the data of the trial if I reject the null or not. So if I reject it or if I fail to reject it. If I reject the null, that means I claim there is a treatment effect. Under the null, of course, that's an error. If there is no treatment effect, but I claim one, that's a type 1 error. And the probability of that error, as we all know, is alpha, which is the significance level that I want to use. The other type of error that I may commit is the exact opposite. There is, in fact, a treatment effect, so I'm under the alternative, but I fail to reject the null, so that's a type 2 error. And in fact, we call this error beta, and as you know, 1 minus beta is the power or the sensitivity of the trial. So under the alternative, I have a type 2 error, or if I do reject the null, I have a certain power to do that. And so in a sense, a clinical trial is a diagnostic tool for the treatment effect, as much as you may have a diagnostic tool for the presence of a disease. It's exactly the same principle. And the frequentist view, as I said, is in fact looking at the sensitivity and the specificity of that diagnostic tool. And we focus on the p-value mostly, which is the probability of the data given the null, right? The probability of observing a treatment effect, that's the data, at least as extreme as that observed if there were no treatment effect. That's the p-value. Under the null, what is the probability of the data? That's the p-value. Now, the Bayesians take a very different approach. They use the same, exactly the same, presentation as before, but they look at the table instead of looking at the rows of this table. They look at the column of the table, which is what now we have some data, And we're going to assume that there is a probability that the null is true, and there is also a probability that the alternative is true. The probabilities, of course, one is, one of these probabilities is one minus the other probability. And so what we want to look at is, given the data, what is the posterior probability that the null is true, and what is the posterior probability that the alternative is true? When I say posterior, that means I have a prior, just like I had before in my exaggeration ratio calculation, and I will show you that I can choose my prior in different ways. And so the Bayesian view is in fact the positive and negative predictive values of the table, and again, it's the posterior probability of the null or the posterior probability of the alternative. Okay, now what about this prior? The prior, in fact, is what you believe the null hypothesis about the null hypothesis, and therefore also about the alternative, before you start doing the experiment. All right, so you may start an experiment and be neutral, and that would be probably what most statisticians would say. You're neutral, that means you have a 50-50 chance that there is an effect or that there is no effect. You don't know, you know nothing, right? That's a state of complete, what shall I say, lack of information. The null is as equally, probable as the alternative. That's a neutral, also called a neutral prior. You may be pessimistic. Again, most statisticians would be either neutral or pessimistic. They would say, well, I don't believe that there is a treatment effect. I really want to reject the null with that belief that it's unlikely to be true. So, it's unlikely to be true. That's a pessimistic prior. An optimistic prior is, well, someone who thinks there is a treatment effect. So there is only 25% chance that there is no treatment effect. And then there is the super optimistic prior, and that's typically the clinician. They are super optimistic because they want to treat patients with a better treatment. So again, no blame. Their bias is in favor of optimism, and that's completely understandable. So they will say, well, if I do the trial, that means I'm pretty sure this new treatment works. Now, sometimes there are good biological reasons to believe that. Sometimes you have so much preclinical data that you will say, well, I'm absolutely convinced this treatment will work. So you may be then an optimistic or a super optimistic person, right? So, I mean, this prior belief is really something that's highly person dependent, or perhaps prior information dependent. Now, let's look at this p-value again. Suppose that you have a neutral prior. Suppose you are a statistician, you don't believe that there's a treatment effect, and you do a phase 2 trial and there is a p-value of 0.09, of 0.05, which is the circle value here. Now, again, the question that I raised before, what is the posterior probability that the null hypothesis is true? Well, this posterior probability is not equal to 0.05, it's actually equal to 0.29, which is much higher. So that means even if you have an observed p-value of 0.05, that does not rule out that there is about a 1 in 3 chances that the null is true, right? So in fact, your level of evidence with a p-value of 0.05, if you have a neutral prior, in fact, this level of evidence is much less impressive than you would think. And that's, again, bad news. And I'm sorry that I'm giving you bad news today, Everardo, but I'm trying to protect you against doing a trial that, you will regret later. And so you will be warned that if you do a trial without thinking, you might have a big disappointment. Now, suppose that I'm a clinician, or you are, I know you're not a super optimistic person, but suppose that this drug is so fantastic. I mean, you've seen the preclinical and you said this is bound to work. So, I mean, I think that, there is 10% chance only that the null hypothesis is true. And therefore, I believe that there's 90% chance that this drug truly works. Again, if you do this experiment and you have the phase 2 and you have a p-value of 0.05, then the posterior probability of H0 is close to 0.05. So it's 0.04, in fact, technically. So in that case, if you are a super optimistic prior, a p-value of 0.05 is enough. Okay? So you have to be super optimistic to believe a p-value of 0.05. Now, the Bayesians go one step further, which I think is quite interesting as well. They look at the odds in favor of HA, the alternative hypothesis, a treatment effect, relative to no treatment effect. So the odds of the alternative as compared with a null. And in fact, this is the formula. Here's the probability of the data under the alternative divided by the probability of the data given the null. And this is called the likelihood ratio. Some people call it a Bayes factor, but it's the same thing, likelihood ratio of Bayes factor. So again, if you have a p-value of 0.05, in fact, the likelihood ratio is at most 2.5. So the odds in favor of the alternative relative to the null are not 19 to 1, I said that before. It's not 0.95 divided by 0.05. It's actually 2.5 divided by 1. So that's a much lower level of evidence. So it's another way of looking at, you know, how a p-value of 0.05 may be uninformative. And it's only if you go to a p-value of 0.005 that the likelihood ratio becomes, you know, pretty impressive, 14 to 1. And if you go all the way to 0.001, then you have a likelihood ratio of 50. So that means the alternative, which is a treatment effect, is 50 times more likely than no treatment effect. And that's good. I mean, if you have a p-value of 0.001, rest assured there is almost surely a treatment effect.
Okay, well, thank you, Mark. Thanks for the warning. So if I understand correctly, we should adjust our observed p-values using a Bayesian mindset to what they actually reflect based on prior expectations, right? Of course, ideally priors that really come before the study or that are adjusted to an individual's, let's say, level of optimism or pessimism. Now, this raises the question of, you know, under a frequentist framework, what p-values should we use? And I was trying to dig this into regulatory documents. I don't think there are many in which p-values are mentioned explicitly, at least not actual values that are required. And there is this, I think it's an update of a former document by FDA, which tells us what kind of evidence is required for approval of drugs in general, not only in oncology. And there are other provisions that I'll discuss later on, but this one is for cases where what they call additional flexibility may be warranted by the agency. And this is specific to the case where there is the so-called unmet need. And in the statistical consideration, they say that typically what is a positive trial is a p-value of less than 0.05, as we all know, two-sided. But in some cases, a lower p-value may be considered, for example, when there is a single trial. And I'll come back to the issue of 1 versus 2 trials. But I find this quite in line with what you said, although it's not explicitly mentioned under a Bayesian framework, or they don't mention what p-values should be used. What do you make of it?
Yeah, sure. I mean, I think the key word here on this slide that you take from a guidance document from FDA, the key word is substantial evidence. And what do you mean by that? And substantial evidence, even though it's not, as you said, it's not explicitly defined in statistical terms, it's actually pretty clear that a p-value of 0.05 is not substantial evidence. And one of the reasons for this that I would like to describe is something that most people don't know. And in fact, it's the fact that if you plan a trial and you design a trial to have good power, high power, to detect a clinically important difference, call it delta, at a significance level of 0.05. Again, I'm interested in the 0.05 significance level here. In fact, if you plan the trial to have 80% power to detect delta, you will actually get to a significant difference, not for delta, but for a smaller value than delta. For, in fact, a fraction of delta, you will actually reach significance. And for example, here, if you have a power of 80%, the value of the treatment effect that will reach significance rather than being delta is in fact 70% of delta. So you might say, well, I mean, that's not what I want the trial to show, because I have an important difference of delta that's clinically important to me. I want to show that. I'm not interested in showing 70% of delta. That's lower than what I've considered to be clinically relevant or clinically important. And yet that's exactly what happens. So see, if you use a 0.05 level, in fact, this factor F here that I consider to be, in a sense, the reduction factor in the effect that you pre-specified, you will see an effect of 70% being statistically significant at a 0.05 level. And so if you really want to detect this effect of delta, then you shouldn't use 0.05. You should, it just so happens that you should use 0.005. for a power of 80%. So you should be much more stringent if you are truly interested in detecting delta or something better than delta. Then in fact, for a power of 80%, you should in fact use a p-value that's far more extreme than what people use. And if you plan the trial to have higher power, paradoxically, this factor F goes smaller. So even if you plan the trial, you know, with a 95% power and you want to use a p-value of 0.005, even in that scenario, you will still have a reduction of your treatment effect that is still statistically significant. That's just, you know, simple stats, but not many people realize that their trial will actually detect a smaller difference than what they had pre-specified.
Yeah, that's really interesting. Okay, so coming back to what I was saying about the FDA document, let's say the major recommendations are about for let's call them usual situations. So what quantity of clinical evidence is needed to establish effectiveness, and so this is the basic case, and so the ideal situation is, as we all know, 2 adequate and well-controlled studies, let's say 2 phase 3 trials in general, but we know that in some cases this is not really what happens. For example, it seems that in oncology, many drugs are approved based on a pivotal study, a single one, and so the FDA says explicitly 1 adequate study that is large and multi-center. So that's, let's say, also acceptable. And yeah, then there are other situations that I'm not planning to go into the details, but I wanted to highlight this because, you know, if I think of it, if you have 1 study with a certain p-value, let's say at the end, or 2 studies with their p-values, what does that, you know, tell us in terms of the evidence that was gathered in 1 study versus 2 studies? What is your view of this?
Yeah. Now, I mean, obviously, there can, you know, there can be differences between the 2 studies. If you do, you know, different studies, you could look at slightly different patient populations or slightly different indications even. So I'm not going to go into that, but if you do 2 identical studies, exactly identical studies in the same centers versus only 1, it's the same thing. I mean, obviously, there can't be any difference whether you split your study in 2 or whether you consider the totality of the study as 1 study. And in fact, if you look at what the FDA requires, they require to have either 2 independent studies at a level of 0.05, or they require 1 study but with a more extreme p-value. And typically they will say, well, you need to have a p-value of 0.0025, which is essentially the same as having 2 independent studies at the level of 0.05. 0.0025 is simply the product of 0.05 times 0.05. And so if you look at this table, what you see here is that if I look again at this factor F, which is the reduction in the predefined clinically worthwhile effect, if you look at the FDA requirement, which is essentially a p-value of 0.0025, you see that that in fact guarantees that the treatment effect will be round about the clinical, you know, important difference, delta, that you have pre-specified. So in fact, it's, you know, sometimes people say, well, the FDA is far too conservative. They want, you know, this ridiculously low level of 0.0025. That's crazy. And that's not crazy at all. I think it makes perfect sense. If you look at it in terms of the effect that will be detected, it is the effect that you were interested in the first place. And you could argue that any lower effect than delta is of no interest. Otherwise, you should have designed your trial to detect that. And so, in fact, the FDA requirement makes a lot of sense from a statistical point of view.
Well, thank you very much for all these explanations. I'm going to try to summarize what you said, Mark, and please correct me if I misunderstood something. But my take-home message from this is that clearly the transition from phase 2 to phase 3 is a dangerous one. It can offer opportunities for bad surprises for many of the, you know, as we saw in many of the examples or the reasons that you explained. And basically these reasons have to do, in addition to the play of chance, to overestimation of treatment effects in phase 2. We tend to be to overestimate what we want to achieve. This is mostly due to the winner's curse or regression to the mean, if you will. There's also the danger of misinterpreting p-values, what they actually mean. And so what can I do about that as someone in drug development? I think I need to correct my expectations, if possible, correct the treatment effect observed in early studies using the framework of the exaggeration ratio, and also consider this sort of Bayesian mindset for decisions and adjustment of expectations regarding the p-value, and also having in mind the issue that you highlighted about the treatment effect, the fact that you can reach significance with the smaller treatment effect that you actually are looking for. Does that summarize your advice to us?
It does absolutely, Everardo. I would just add something that is not statistical, so it's more in your court, but I think the advice I would give to people when they plan phase 2 designs is to actually use a sensitive endpoint, to not be misled by p-values or to not miss an effect that might be relevant, to use as sensitive an endpoint as possible. And that's probably not response rate, it's probably not PFS, it is certainly not OS. And so I think a lot more thinking would need to go into the endpoint that we use, as well as to these effects that we have discussed today about the statistical significance.
Okay, good topic for a future webinar, huh?
Absolutely. Yeah.
Okay. All right. So Kelsey, I guess we can hand it back to you, and we're ready for the Q&A if we have questions from the audience.
Yeah, absolutely. And thank you both so much for the presentation. It was really informative as always, and I'm sure the audience enjoyed it as well. And just a reminder to our audience, you can still submit questions in the questions panel. So please do if you have any. And we'll go ahead and start with the first couple that we've received, if that works for you. So
Sure.
the first question that we have is, when would you recommend using an optimistic prior versus a neutral one in their exercise of computing the posterior probability of the null hypothesis? And could data from phase 1 trials be informative?
Is that for you, Everardo? Because you are the optimist, right?
You know me, right, Mark?
Because my answer would be never be optimistic. I mean, that's against statistics, right? We want to be pessimistic and then have good surprises.
Yeah, it's better policy. Yeah, but the question about whether phase 1 can help is a good one, huh? How can we use that?
Well, I mean, when a phase 3 trial fails, it's not uncommon for sponsors to come to, to us and to other statisticians and to say, well, how do you explain this? I mean, it's impossible. It's, what went wrong? We use the same centers, the same patients, the same everything. The phase 2 was identical to the phase 3. And why are we so disappointed? I hope we have addressed this in part today. Certainly, that's on average what we would expect, that there is disappointment in phase 3, at least small, to a small extent. It may not explain fully the sort of huge differences you've shown in your slides, where you had a wonderful phase 2, with a huge separation of the survival curves, and then nothing at all in phase 3. That is too extreme to be explained just by a statistical regression to the mean. And those cases happen. Now, again, we have a biased sample, because the reason we've chosen these examples is precisely that they are very extreme.
Yeah.
So I think we always need to worry about whether we have a biased sample, even for examples that we've used in this talk.
Okay, Kelsey, do you have other questions?
Yes, we do. Let's see, next question. Wouldn't a highly significant randomized phase 2 trial compromise enrollment to a follow-up phase 3?
Well, that's a good one. I guess, yeah, the question is, yeah, if you have an observed result in phase 2 that is, let's say, a very low p-value, what is people's motivation to move forward? I guess that's the question. Well, I know, Mark, your take on non-randomized trials, right? But what can can having a randomized phase 2 trial with very good results backfire in terms of your ability to conduct a phase 3 trial, do you think? Either ethically or because people will not want to take
Well,
the
I mean,
risk?
ethically, that's not an issue for me to discuss, but I think you are only ethically allowed not to do a randomized trial if you know that the treatment works for sure, or nearly for sure. And if we do a randomized phase 3, that means by definition that you're not completely convinced by the results of a phase 2. And as I've shown, the level of evidence that the agencies require is pretty high. It's not just a phase 2, very good result. You need to confirm this. And I mean, the agencies believe in, the need to replicate. And I think they're right. We see so many other scientific fields where replication is not required. And in fact, people believe a false result simply because it looks nice and it's so nice to believe in it. I think the agencies, to be honest, I think, I'm glad as a patient that we have the FDA and the EMA and the other agencies, because they make sure that a treatment that goes on the market is truly effective and safe. So, I mean, it's obvious to say this, but I think it needs to be repeated. And the level of evidence they require is not too high. If anything, the purpose of this webinar was to show it's not too high. It's what we need. It's not higher than that.
Right. Okay.
But I think the problem, perhaps, of the point of the question was to worry about accelerated approval, which we haven't really discussed today. But if you have really outstanding phase 2 results, the FDA may grant you accelerated approval. And that is it's a good mechanism because it allows patients to access drugs early, but it can backfire because it makes then the phase 3 trials much more challenging. If the drug is on the market, some patients will not want to go into a controlled trials because they will say, well, the drug was approved. Why should I have, the risk of going to a placebo arm or a control arm? So that's kind of probably a perverse, a perverse side effects of accelerated approval. I think in many cases, accelerated approval is justified and a good feature, but it can have sometimes negative effects. And in fact, accelerated approval, as you know, Everardo, is only granted if your phase 3 trials are, either ongoing or at least planned. And
yeah.
And because again, the need for confirmation exists still.
Yeah.
It becomes perhaps more difficult in those cases.
Yeah, it's good that you mentioned this because we looked this up recently, and it's written explicitly that the phase 3 should be on the way, at least preferably. Yeah. Okay, Kelsey.
All right, next question. So, does exaggeration ratio depend only on the magnitude of the estimate, or will it be influenced by design, sample size, or other elements?
No, it depends, as I showed. This is the beauty of it. It depends only on the confidence interval of the estimated effect. As I showed, if you look at the website, and the slides will be shared so people can actually go on that website and play with it. It's quite fun to do that. If you know the confidence interval of your treatment effect in the phase 2 or in whatever experiment, you can actually calculate an exaggeration ratio. Where I think it becomes a bit difficult is that the database of all trials, that Cochrane database of trials, may not be a very relevant source because it contains trials of all nature. The comparative trials, okay, but on many indications. And some people will probably want to use the same approach, but on a better basis of trials, perhaps in that same indication, or some such approach, which would be more focused on the problem that they have.
Great. All right, we'll get another couple more questions in if we can. So one of these here, if a subgroup analysis in a phase 2 trial has been positive, but it's a subgroup with a small n, should you do another phase 2 trial, for example, a phase 2b, before a larger phase 3?
Sure, I would definitely recommend that, or perhaps have a seamless phase 2/3 or a phase 3 with a early interim analysis. I mean, something that really allows you to pull the plug if you see that you're not confirming this result, which is most likely false. I mean, let's face it, a subset result with a small n, I can't believe that is, going to convince anybody, even the sponsors.
Okay, and I think we have time for one more question here. What do you recommend as adjustment of expectations when the phase 2 evidence is based on a single arm trials? Can the overall response rate be used in that case?
That's more for you, Eduardo.
I don't know. I mean, of course, we know that in oncology, you know, where this has been looked at. Let me start again. So obviously, the overall response rate is a very good, let's say, screening endpoint for new drugs, obviously. But we know that it has little correlation with long-term outcomes, particularly with overall survival, where this has been looked at formally. So, you know, one part of the question is, how is the overall response rate useful? Not very much. The other part, it's beyond my knowledge to say, can this be used in a way to correct? I mean, can you play with the expectation? The exaggeration ratio by, let's say, converting your response rate to some expected, you know, relative reduction in another endpoint, Mark? I think that would be a leap of faith, right?
Yeah. I mean, yeah. If you don't have a proper control, you just can't do this reliably.
Yeah. Well, even suppose you have now a randomized phase 2 trial and you have the odds ratio for response. Could you play with the odds ratio?
Oh yeah, you could do that if you have a randomized. But I think the question was about single arm phase 2. Single arm. I mean, that's a different ball game. I won't even say anything about that.
Yeah, because of what you said, right? The risk of selection bias. You never know.
Yeah, I mean, sure, but I mean, the fact is that sometimes you do a phase 2, randomized or not, and you have such an outstanding response rate that, you know, this is really something. I mean, you know that you're on, and then the responses last a long time. So I'm not denying that in these situations, you can run to accelerated approval because it shows an effect that's completely beyond the random play of chance. But however, as you said, then you still have to show that there is a clinical benefit to these patients in some way. So a confirmatory trial, you know, still needs to be done. But this is probably the situation that most people have in mind. You know, what if I have an outstanding response rate? Well, that's of course wonderful and very good to have, but it's not the end of the story by any means anyway.
Great, and I see we're at time. So thank you so much to the both of you. That was a great presentation, Dr. Busey and Dr. Saad. And thank you for our audience for participating. Just a quick note, you can submit questions if you have anything in addition to what you've submitted today to info@iddi.com. So please feel free to send questions in afterwards. And again, thank you so much for attending. We hope to see you at the next webinar. Thank you both.
Thanks, Kelsey. Thanks, Mark.
Thank you.
Thank you, Eduardo. Bye-bye.
Bye.